All right, we are on to what should be our final section in the lecture on the digestive system part of our chapter. And so the last little bit of this section here is going to look at how gastrointestinal activities are controlled and coordinated by both the nervous system and the endocrine system. So what actually controls gastric motility and what actually controls release of digestive enzymes, those sorts of things is the focus of this last section here. Typically, we've seen before, when we talked about the salivary glands earlier in the chapter, we talked about how simply even having the sight or smell or even thought of food can stimulate salivation, and that is the case. The nervous system is very, very adept at getting ready for the possibility of food coming into the digestive system. So gastric secretions, some of the enzymes, some of the hydrochloric acid and so forth secreted in the stomach, those will be secreted even before you take that first bite of food in anticipation of food actually hitting the digestive tract. Proteins, for example, if proteins arrive in the stomach, there is going to be an immediate release of gastrin. Gastrin is a substance that's going to help increase the production of both HCl and pepsinogen. And if you think about what HCl and pepsinogen do, both of those, the acid as well as the enzyme, are what eventually turns into pepsin from the pepsinogen. Both of those substances are involved in digesting and breaking down protein. So it makes sense. You've got the arrival of proteins. That's going to trigger gastrin as a hormone. Gastrin is then going to trigger HCl and pepsinogen, which are going to start breaking down that protein in the stomach as quickly as possible. So it's kind of a logical progression of events that takes place with protein digestion. There's also a whole host of what are called enterogastrones or duodenal hormones that are involved in helping to slow down the digestive process in the stomach. And so you might say initially, well, why in the world would you want digestion to slow down? Wouldn't you want it to go as quickly as possible? Well, not necessarily, because once food goes into the duodenum, right, once food reaches the small intestine, it does take a bit of time for those enzymes coming in from the pancreas, the bile coming in from the gallbladder, the bicarbonate buffer. It takes time for all those substances to do what they need to do in terms of breaking down the remnants of your meal. And in addition to that, you also have to absorb the parts that you need to absorb. So when you go to absorb the amino acids, the monosaccharides, the fatty acids, that takes time as well. So if you simply rushed everything through the intestine too quickly, you wouldn't actually be taking full advantage of all the nutrients that you're consuming. So what happens here with these hormones, we've got a group of 3 of them here, and a couple of them at least are kind of a mouthful to say. 3 of these are involved in helping to make sure that the small intestine, especially the duodenum, has enough time to not only break down, but also absorb the nutrients from the meal that you've consumed. So we've got 3 of them here. We've got one that's called cholecystokinin, which is also called CCK, mercifully much shorter, much easier to say. We've got secretin, and we've got one that's called gastric inhibitory peptide, and that's one that's going to be involved with, especially with stomach motility and secretions of substances there. So we've got CCK, secretin, and GIP. CCK is involved in stimulating the gallbladder, as well as the pancreas to release the bile and pancreatic enzymes, respectively. CCK is going to cause those to come in. When CCK is increased, so too is bile and pancreatic enzyme secretion increased. Secretin is involved in helping to allow for more bicarbonate buffer to come in. Remember, think about what that does. That's involved with neutralizing the pH of the chyme coming in from the stomach because it was initially very acidic, right? So we want to neutralize that if possible. So here's kind of this is a mess, isn't it? This is a flow chart, one of those negative feedback sort of a things that we saw before, but I'm going to kind of walk you through it. And this is, again, a really good chart. If you can sort of walk your way through this, explain this to somebody else, you really have this down pretty well. So what we have up here, this tiny little thing back here, this is the esophagus. Right? So we've got food coming down the esophagus into the stomach. And so immediately, once this food hits the stomach, chances are it's going to be much less acidic from the stomach environment, so the pH of the stomach is going to go up. And in addition, with the arrival of proteins in the stomach, that's going to trigger a release of gastrin. And what gastrin does, like we talked about on the last slide, gastrin is going to help increase the production of both pepsinogen and pepsin, as well as HCl, and those are going to start helping to break down the proteins that have arrived in the stomach. Okay, so from there we're going to move from the stomach, kind of back behind the pancreas, into the duodenum of the small intestine. Once we have arrival of material there, we now have the release of gastric inhibitory peptide. So this hormone here, this GIP, is going to help decrease contractions of stomach smooth muscle lining. The muscularis of the stomach is going to slow down in response to GIP being secreted by the duodenum. We're also going to have a decrease in production of our 2 substances, our pepsinogen to pepsin and our HCl. So what that's going to do, both of those two things together, decreasing stomach motility and decreasing stomach enzyme and acid production, is going to help slow down what the stomach is doing for digestion. And that's going to help give the small intestine time to break down and absorb the materials that it needs to deal with. Meanwhile, in the small intestine, we have the arrival of more materials here, so we're going to have a secretion of secretin. Secretin is going to feed back over here. We follow this line back to the pancreas, and we see that secretin causes bicarbonate buffer production to increase in the pancreas to help neutralize the environment of the small intestine because it was very acidic with the arrival of the chyme coming in from the stomach. We also have CCK, right? CCK is going to cause the gallbladder to produce bile to help emulsify fats coming into the small intestine. And CCK is also going to, if we follow this line down here, it's going to feed back to the pancreas and tell the pancreas to start secreting enzymes. So secretin causes bicarbonate buffer production to go up. CCK causes both bile and pancreatic enzyme production to go up. So again, you go back to your proteases, your lipases, and your amylase. All those substances will increase from the pancreas once CCK is produced. So at the beginning, it looks kind of complicated. It is fairly complex, but if you sort of take it step by step and think about what all these terms are, if you know this really well, you should be able to put it together into one kind of big cohesive picture. We've seen this concept before, so we're not going to spend a ton of time on it, but looking back again one more time at blood glucose, you have a really carbohydrate-rich meal, your blood glucose goes up. So if we look at carbohydrate-rich meal, increasing blood glucose, in the islets of Langerhans in the pancreas, the beta cells are going to increase their insulin secretion, and the alpha cells are going to decrease their glucagon secretion. And so those two things combined are going to help form glycogen in the liver and fat in the adipose tissue, and both of those two effects together are going to help decrease blood glucose. If you have either a fasting situation or if you have a situation where you're exercising heavily and burning up a lot of glucose, your blood glucose goes down. So that's going to trigger now in the pancreas the alpha cells to produce more glucagon and the beta cells to produce less insulin. And so both of those two things together are going to help release glycogen from liver cells and muscle cells, and also help release calories from the fat that you have stored up also in adipose tissue. So we have that one as well. We also have the possibility, what we just talked about actually is called glycogenolysis, which is where you take glycogen and break it down to release glucose into the blood to raise blood glucose levels. The other option we can do is called gluconeogenesis, which is where the liver takes other things like fats and like proteins and turn those into glucose or something like glucose to allow you to continue to burn calories and fuel the production of muscle contraction and so forth in skeletal muscles. So we've got the ingestion of food doing two main things. It's energy, it's calories. It's also building blocks, right? So if your body needs to make proteins, if your body needs to produce fats or steroid hormones, if your body needs to produce glycogen, you got to have the materials to do that. You're not creating materials out of nothing. So if we look at what's called basal metabolic rate, if you're just in a resting situation where you're not exercising, you're not burning a ton of calories, you're barely doing more than just sitting in your chair and breathing, right? Not even thinking very hard, right? The number of calories you burn during that time is called your BMR, your basal metabolic rate. If you consume more calories than you burn, then clearly those calories are going to be stored up. And so we worry about fat, for example, accumulating as a result of overconsumption of excess calories. The body is not built to do anything except store excess calories, because throughout most of our evolutionary history, our ancestors didn't have a ton of excess food or simple carbohydrates, simple sugars to consume. Whatever calories they found, who knows when the next time they would find another meal might be, so they would store those calories. Their bodies were very well equipped to not throw anything out, to store calories and save them for a rainy day, if you want to use that kind of expression. And because we're descended from our ancestors, our bodies do the same thing. Our bodies don't know that they shouldn't consume excess calories, and since our behaviors lead us to often consume more calories than we should, or often lead us to burn fewer calories than we should if we don't exercise enough, our bodies still do the same thing as our ancestors. They store the calories up, and of course that can lead to health as well as aesthetic problems when it comes to storing excess energy in our bodies. So we've seen before in our chapter on the endocrine system, we talked about leptin. Leptin's a protein hormone. It's one of the things we think that maintains a certain degree of appetite control for us. But again, in humans at least, feeding behavior is not just related to hunger. Most of us eat all the time when we're not actually hungry. You hear somebody say that they're starving. Well, that's probably the farthest thing from the truth. They maybe haven't eaten for a couple of hours, but I guarantee you they're not starving. So we eat for a lot of reasons, aside from just hunger and aside just from the control that hormones might have over our bodies. Leptin actually is produced by the adipose tissue, so fat cells, when the body is accumulated enough calories in a certain meal, adipose tissue starts to produce leptin, which then feeds back and tells the brain, in sort of kind of an indirect way, that, hey, you should probably stop consuming food now. The receptor for this one is in the hypothalamus. So again, we have the situation where hormones are produced by adipose tissue, and the actual receiver for them isn't until you get to the hypothalamus, which then can cause changes in feeding behavior as a result, telling the brain, like I said before, to stop taking in food. Some of the other things we talked about before, right? So the insulin, gastric inhibitory peptide, cholecystokinin, all three of those are also involved in stimulating to the brain that you've had enough to eat. In other words, they tell you that you are satiated and you can go ahead and stop feeding now. You can go ahead and do other activities that might be productive for you. One called ghrelin is kind of the opposite. It's an antagonist to the other three, so it's one that stimulates food intake. So if we look here at the hypothalamus, right, we've got all different kinds of control over feeding behavior. Ghrelin has a positive effect on feeding behavior. Gastric inhibitory peptide, cholecystokinin, insulin, leptin from adipose tissue, all three of these have a negative effect on feeding behavior. So you see some of the effects that can occur as a result kind of indirectly from this. These are all signals going into the hypothalamus. So we say these are afferent signals traveling to the hypothalamus, whereas efferent means these are things that occur as a result of what's going on in the hypothalamus. These are messages or activities that are being sent from the hypothalamus. So we look at things like appetite and feeding, reproduction and growth, energy expenditure, and so forth. All of those things are affected by what the hypothalamus perceives the balance to be between things like the inhibitory versus the stimulatory effects of these different hormones. There's also one called neuropeptide Y, which also helps to induce feeding activity, kind of like ghrelin does. It's sort of the antagonist to the others, the leptin, insulin, and GIP/CCK. MSH, we've seen MSH before, melanocyte-stimulating hormone, which normally causes melanocytes in the skin to produce melanin and make your skin get darker in response to UV light. This is one that has an antagonistic effect on the neuropeptide Y. So we see like multiple levels of control here. We've got neuropeptide Y inducing feeding activity, MSH suppressing neuropeptide Y, so it's suppressing feeding activity. It all gets pretty complicated once you start looking at the chemical aspects of this control. As far as nutrients that we look at, a lot of the things that our bodies need, we can't produce for ourselves. So in the animal kingdom, for example, there are all kinds of different species that, for whatever reason, can't make certain things. So if you look at primates, of which we're that, humans are primates, and also guinea pigs of all things, cannot produce their own vitamin C. Most other animals can produce vitamin C, but our two groups, the guinea pigs and the primates, ancestors, for whatever reason, a mutation somewhere along the lines caused us to fail to be able to produce vitamin C, so of course we have to get that in our diet. For that reason, vitamin C would be what you consider an essential nutrient. Essential nutrients are things that you have to get in your diet because your body can't produce it for itself. Other things we can produce for ourselves. If we look at the essential amino acids, there are 9 of them that we cannot produce for ourselves. And it turns out it's not just humans, it's pretty much all vertebrate animals that trace our ancestry back to fishes. We all have the same 9 amino acids that we cannot synthesize for ourselves, so therefore we have to consume them in our diets. Those are what we call essential amino acids. There are some other long-chain unsaturated fatty acids we cannot consume for ourselves. Those are ones we have to, we can't build for ourselves. We have to consume them in our diets. We can't build them ourselves. We can make cholesterol in our bodies, which is kind of a handy thing to be able to do. Some insects can't. There are groups of things that cannot make some of the same things we can. There are some things that can make some of the things that we cannot. So it varies by group in terms of which sorts of things might be essential versus able to be made in the body itself. Minerals are inorganic things. You look at things like iron and magnesium and calcium. Those minerals are also ones, obviously, you have to consume for yourself because your body can't manufacture those. All right, so that's the end of our section on the digestive system. What we'll do is we'll get this closed up and we will start on the urinary system.